The last decade of the 20th century began with one of the most ambitious scientific undertakings of all time. The Human Genome Project promised to decipher our genetic code. One prospect was that by identifying genes responsible for disease, we could develop treatment. Yet each discovery confronted a sobering reality. Because we know a specific gene leads to a particular illness, doesn't mean we can do anything to stop it. However, University of California, Berkeley geneticist Jennifer Doudna plans to change that. According to the LA Times of February 1st, 2016, Doudna's gene editing technique, called CRISPR, can be used in a living organism to detect any gene, remove every copy of it, and replace them with new ones. These permanent alterations are then passed down to future generations. Traditionally, genetic manipulation has been reserved for specialists in a billion-dollar lab. This method can be mastered by grad students for less than $1,000. The Genetic Literacy Project of February 6, 2016 explains, editing DNA will now be quick, cheap, and far too easy. Doudna admits CRISPR could eradicate disease, but should probably be banned on humans. So let's first explore how CRISPR works. Next, analyze applications and obstacles, before finally uncovering implications. To the method that Harvard genetics professor George Church explains, could be as important to this century as vaccines were to the last. The Human Genome Project identified the building blocks of a human being. CRISPR allows scientists to deconstruct and reorganize those blocks. To understand how, let's first look at what CRISPR is and how it works. First, when studying DNA from various bacteria, Doudna noticed something peculiar. Spliced throughout every bacteria's DNA were small bits of code that didn't belong. Upon further investigation, Doudna realized that these bits of DNA are from a virus. NPR explains on June 6, 2015, when bacteria encounter a virus and survive, bits of the defeated pathogen are then incorporated into the bacteria's DNA. Think of it like a wanted poster. When the virus strikes again, the bacteria recognizes it and releases an enzyme Doudna calls molecular scissors. I've taken some artistic liberties. Which enter the virus and cut that part out. Next, Doudna believed this defense system could be harnessed offensively. Wired explains in July of 2015, by changing the wanted poster, you can create molecular scissors to hunt down anything. Let's say the virus DNA is replaced with the gene responsible for breast cancer. When exposed to a cell containing that gene, the resulting molecular scissors will hone in on every copy and snip them out. Essentially, whatever you put here, CRISPR will remove. Moreover, Doudna tells the previously cited NPR, if you place a new, healthy gene near the break, repair enzymes pull it in, replacing the faulty one. Like Control F finds words in a document, CRISPR detects every copy of a gene, deletes, and replaces them. The Human Genome Project identified hundreds of genetic markers that predispose us to disease. CRISPR allows us to do something with that knowledge. Let's explore possible applications and looming obstacles. First, CRISPR has the potential to treat disease. The previously cited Wired details how researchers are using this technique in mice to reverse mutations that cause blindness and stop cancer cells from multiplying. According to Science Reports of March 4th, 2016, scientists have even removed HIV from human cells and made them immune to future infection. These alterations are not limited to the recipient. If someone has the Huntington's gene, CRISPR keeps that mutation from being passed on to a child. And this process could begin in the womb. Business Insider reveals on August 4th, 2015, CRISPR could engineer an embryo for any genetic predisposition, be it weight, height, or IQ. Next, scientists have urged caution when it comes to human testing. Physics.org explains on February 6, 2016, CRISPR sometimes produces insertions and deletions in unintended regions of the genome, leading to unexpected results. And while accuracy is steadily improving, a mistake in a human embryo would be risky. Genes often work in tandem, meaning removing one could produce unpredictable mutations. Instead of treating a disease, you could create one that's undetectable until birth and inheritable. In fact, in a March 12, 2015 Nature article, a group of scientists called for a ban on CRISPR's use in human embryos, calling it dangerous and ethically unacceptable. Today, we are tasked with how to apply the knowledge attained by the Human Genome Project. CRISPR yields fascinating implications concerning ethics, genetics, and gender. First, CRISPR exacerbates an ethical divide between the West and China. 37 days after the Nature article called for a ban, a group of Chinese scientists revealed that they had used CRISPR to modify human embryos. The May 2015 edition of the journal Protein and Cell explains, of the 71 embryos, CRISPR replaced DNA in 4. None were brought to term. National Geographic of April 22, 2015 contextualizes these seemingly unimpressive results, comparing them to early work with cloning. Clumsy, but inevitable. Despite widespread disapproval, the New York Times of June 29, 2015 explains, the scientists intend to keep trying. While we engage in broad ethical discussions, others won't be guided by Western principles and plan on exploiting CRISPR's full potential now. Next, as we get better at snipping out problematic genes, where will the replacements come from? Bloomberg explains on June 29, 2015, drug companies like Amgen and Genentech are currently collecting and patenting uncommon genetic deviations from all over the world. For example, the DNA of a man with bones so dense, they don't break, and genes that make skeletal muscles 40% stronger. Stem cell expert George Daly tells The Verge of April 23, 2015, as we continue to realize CRISPR's full potential, it raises fundamental questions about who controls our genomic destiny. Some corporations have already decided. With CRISPR, rare mutations become valuable commodities. Finally, Jennifer Doudna's role in CRISPR's discovery has encountered resistance. Jezebel explains on January 20th, 2016, she was the first to discover CRISPR and submit a research paper. Meanwhile, MIT geneticist Feng Zhang submitted his CRISPR research weeks later, but paid to have it fast-track published 2 weeks before Doudna's article. And this past January, the journal Cell published his definitive history of CRISPR without consulting or even mentioning Doudna. Jezebel continues, an institutionally sexist research community all too often determines who gets their name in textbooks. In the tradition of Lise Meitner's discovery of nuclear fission and Rosalind Franklin's discovery of DNA, Doudna may soon join an esteemed list of pioneering female scientists, all written out of their narrative. When Doudna first stumbled across a bacterial defense mechanism, she had no idea it could change the world. After exploring how CRISPR works, its applications and obstacles and implications, we now have a better understanding of how it might. Though the Human Genome Project technically ended in 2003, with CRISPR, its impact continues today.